Alias in the Hyderabad region, so it's my humble request to both of my scholars just focus on your and objective and clear the important finding in the audience. Okay, so over to Yousra.
Thank you. Bismillahirrahmanirrahim. Assalamu alaikum to all my respected teachers and my dear. This is Yousra and I am going to present my final M.Phil seminar under the supervision of Professor Dr. Riffat Sultana. Now let's move towards the title of my research topic, that is behavioral study of Acheta domesticus under controlled conditions. Here are the contents of my research. Introduction, objectives. Here are my objectives: to survey the various localities of Hyderabad in order to collect the tragedy, to investigate the feeding habits and mating behavior of Acheta domesticus under controlled conditions, and to analyze the locomotion pattern and the social interaction to understand their behavioral dynamics, to examine aggression and communication behavior under controlled conditions. Here are the material and method: cultivated fields, roadside areas and moist areas. Sampling techniques. The stock of insects was collected by hand picking method. To get the maximum collection, the activity time was being noticed, and in one trip, twice the sampling was collected. First in the morning to dusk, and second 2 to 3 hours after the sunset. This is figure number 2 showing the field survey. This is the standardized ecological method which I use. Some of the insects reared in the cages, then the killing of some specimens were being done with the help of chloroform, and the preservation and examination of my specimens was being done. These are the laboratory observations. All the laboratory observations were carried out at the ecological and biocontrol station in cages. Here are the living cages and jars for the sort. The cages and jars were kept in the research laboratory where the temperature varied from 33 to 38 degrees and the relative humidity measured from 0.6 to 71%. Field condition had a similar temperature and relative humidity, which is only found in the case of field. Killing and preservation. Specimens were killed with the help of chloroform. They were shifted to the stretching mold. When they become fully dried, they were shifted to the insect boxes, and the identification and naming to the specimens were being done. Here, now we are moving towards the result section of my research. Here we are discussing about the systematic position of Acheta domesticus. Kingdom Animalia, phylum Arthropoda, class Insecta, order Orthotra, family Rallidae, genus Acheta, and species. The dorsal and lateral view of male and female Acheta domesticus. This is the table number 1 showing the measurements of various parts of Acheta. Experiment on optimal of Acheta domesticus. So in this table, we are presenting the rearing density and their effect on the survival rate and the growth. So here is the rearing density. In one cage, we kept 19 specimens. In the other, we kept 47 specimens. And in the last, we kept 93 specimens. And we saw that what is reduced fertility recorded for 47%. The number of specimens collected was 1,000. This is figure number 8, showing the survival rate of vegetable and the environmental condition. These stages of the mating behavior are calling and courtship, antennal contact, mating, and post-mating behavior. The life cycle of Acheta domesticus. They were given with different diets under जी
मैं भी बोला था ना ये सब कुछ पता है